Now, what if 2 years after the birth of your son, your doctor gives you devastating news that soon your son will be unable to keep up with his friends? He'll be unable to get up quickly by himself. What if when he comes into his teens, he has to get off his feet and go onto a wheelchair? Later on, he won't be able to feed himself, wash his own face, even give you a hug. And what if by his 20s, he has to go on respiratory support and have invasive spinal surgery only to die in his mid-20s out of respiratory insufficiency or cardiac failure? Now, what this means is that your life does not have outdoor activities. There's no Little League, and even simple things like weekly family outings just become a huge challenge. Now, this tragedy is a reality for many, many parents whose sons have been diagnosed with Duchenne muscular dystrophy. Duchenne muscular dystrophy is the most common genetic fatal childhood disease. And with 1/3 of the responsible mutations occurring spontaneously, it's not a disease that'll just go away by genetic counseling. Now, Duchenne muscular dystrophy is caused by an absence of this protein dystrophin, which is like a shock absorber. So during contraction, your body sustains a certain amount of damage, and dystrophin helps protect your body from that damage. Now, in Duchenne, while the muscle develops normally, it's easily damaged without this protein. And initially, this damage is mediated by these endogenous muscle stem cells populations. But after rapid turnover, this population is exhausted. Interestingly, this stem cell population, like other stem cell populations, is located very close to blood vessels. And from our work, we've shown that if you genetically increase the amount of blood vessels in a mouse afflicted with DMD, you can actually mitigate some of the damage and even increase the lifespan in these mice. Now, my research is twofold. First, I would like to look at the interactions that muscle stem cells, and really other stem cells that are really close to the blood vessels, and what they have with the blood vessels, and what's mediating those interactions. My second goal is a more translational goal, to see if we can use small molecules or antibodies to increase angiogenesis so that we can potentially copy some of the things that we see in our genetic models. Now, with my research, hopefully, we'll not only be able to figure out crosstalks between stem cells and blood vessel cells, but also come up with novel vascular targeted therapy for Duchenne muscular dystrophy. Thank you.